fourth part. And
in between, there will be a break.
There will be a break in between, 1
Yes.
hour or so. So to make you not so exhausted after that. But anyway, thank you so much, Professor Heike. Please continue the explanation.
Yeah, thank you. Thank you. Actually, then also giving me the chance to help you in this way, because I think it is an important methodology whenever you want to do monitoring in the long run. If you want to identify what you have in the field, molecular data are absolutely necessary in the future. So it's this issue. My course is only about the methodology, but of course you also need to include more experts on the various groups. We do not have the experts in Germany on all taxonomic groups in coral reefs. This is impossible, but we have some. We can address them then also in the future for certain groups. So as I am the expert on marine heterobranics, but not for anything else. Okay, so that is the last talk. And now today we want to know how to create a phylogenetic tree. And first we have to ask, why do we need a tree? Why do we create trees and what can we do with them? There are many interesting subjects and topics that we can do with the trees. I think you remember that I was, wait a minute, I have to do this a little bit different here. I showed you already this picture in the very first lecture, in the first seminar, where I showed you that we made a tree. This is the full tree. It's not rectangular. It's in a circle because there are so many taxa in here, so many species listed here. And to show it a little bit on the one page, this is a way to how to present a tree. It's still a tree. And then you can see that we have here, for example, the most basal groups here, and then we have the next related ones, and that's actually on the top of the tree here. And the questions that we were trying to understand was more in evolutionary issues about how the incorporation of chloroplasts have evolved. But if you want to work on these things, you need to work internationally with many, many different species from all over the world. So in general, actually, what you are doing more and probably in Indonesia when you start is you want to know more about the species, you want to know more about the populations, a little bit about the higher taxa where it belongs to. And then the further things, what I just said, are these biological phenomena to understand how they evolved, or parallel or joint evolution of characters. So this is, but we always have to keep in mind that this is only a hypothesis. A tree is only a hypothesis. We do not know whether this tree is really true. Because, as I said, this evolution of the species and specimens has happened in the past. We were not there. We can only provide evidences that this tree might be, correct. And these evidences are different kind of genes. It can be morphology, but I have talked already about this. Oh, it's frozen. I have to wait a second. No, okay. Here I actually want to show you the issue that is probably of most interesting for you. And I take now this publication from Adele that she has provided or done in my group on CISLA group on the Phylidiidae. I have showed you already the full tree of these Phylidiidae, and you see there are about 700 or 800 sequences of specimens that were here included. I show you just the detail, just this one here, and then you see that we have several specimens, could identify them to a certain species. And the way we are doing this is actually by including our own sequences and combining them with sequences from repositories. So I have here, actually these were our own sequences. We have here our identifier here. And then we also included here sequences from the repositories here. That's so the accession number from the from NCBI, from this repository gene bank. And then when we see what actually groups together, we have here a long branch, very short branches inside, means that this is very well supported by a lot of mutations, a lot of genetic distance. And then we can say this really belongs to a certain taxon, and in this case we could name it with Phyllodeopsis cricensis. So even if we have not correctly identified one or the other specimen here, we still might be able to find where these animals belong to, and then can also check the morphology again. However, we rely, of course, a little bit to a correct identification, that we are not completely out of the range here. And we also rely to a hopefully correct identification of the specimens that we are now in gene bank, that this, for example, and this one, and this one, and this one are really Phyllodeopsis cricensis. And I can only tell you that we have found several cases where this is not true, that the identification of a species, the identification of a certain specimen was not correct and did not belong to the group that it was meant to. But this, to find out, also needs a little bit of training. Okay, so the alignment, just now starting with this, we have to have a good alignment. The alignment is the most important part for all subsequent analysis. Whatever you do, whether you do population genetics, whether you do really phylogeny on understanding evolution, when you have an alignment that is not very well done, you have, of course, a problem in all the subsequent analysis. So the prerequisites for a tree reconstruction is first that you have a proper quality check of your sequences. That means when you have performed your own sequencing, have collected your own material, ask the company to do it or ask the colleague to do the sequences, to obtain the sequences, you have to have really a nice quality check, a good quality check, so that the sequences are okay. And then when you download the sequences, include your own sequences, you have to look that your alignment has a high quality, meaning that you use a really good system like MAFT, what I have shown you last week. And also you have to check the alignment again, because there might still be mistakes in there. Also, the alignment programs make mistakes. Next problem when you do a tree reconstruction is maybe that the genes that you have selected are too conservative, meaning that there is no information in this. So I just show you here now a selection again of adults data set here. And you see these gaps, they do not count, of course, that we cannot use, but we do not have a lot of mutations here. We have one here in this specimen and another one here. And there's only one thing that really unites a group. And when you have a situation like this, the computer programs can hardly decide who is more closely related to each other. And this then provides you a tree that actually looks not like a tree, it's more like a comb. Yeah, so it would look like this. In this case, it would look like this. And only these 4 sequences here that have a uniting character can be analyzed or can be judged by the calculations, by the analysis as maybe belonging together. But when you have a situation like this through the whole alignment, this is only a small part, of course, but when you have a situation like this through the whole alignment, you have to go for a different gene, because then the gene cannot provide you a tree. It's it will always make it like this in some form, but by the length of the distances, you can see that it is actually a comb. I'm sorry. So this is one problem in the tree reconstruction, that you have no mutations. It could also be that there are too many, that this actually is so so confusing, then the algorithms can also not decide what is belonging to what. That's also not really good. Then another problem is that these mutations are not evenly distributed with regard to the taxa or with regard to the genes. And this happens very often when you are working with several genes. You remember I told you that we have concatenated last week, we concatenated CO1 we concatenated this with 16S and H3, and they all have a different weight of mutations. So the mutations are then not evenly distributed. And some of the elder computer programs could not deal with it, but the new ones can do it. And I will explain also now why. Another problem is, and I addressed this already last time, is when you have a lot of missing data. Yeah, when you have when you have too many gaps or too many question marks, then of course the computer cannot tell what belongs to what. So in this case, it has information on this gene for these species here, and then it has another information on these species. So how can the computer then know which of these ones is more related to these ones? This is impossible. So when you do a concatenation, you need overlapping. You need a lot of overlapping information between the different genes that you are using. Now, we have to talk a little bit about the 4 kind of mutations that can happen, and about evolutionary mutation weights. And you might remember that I was telling you about these 4 different nucleotides that are forming up the helix. We have here the guanine and the cytosine, and the guanine and cytosine can combine or do combine with the 3 bonding system here, whereas adenine and thymine have these 2 bonding systems. And I said already that adenine and guanine can be exchanged more easily because of these similar constructions here, and thymine and cytosine can more easily exchange and mutate. So we have to consider now the mutations with regard to these 4 available nucleotides, because it's not happening that only this is changing from adenine from guanine. It can also be that thymine and cytosine are also, so that the thymine and guanine are also exchanging. But this, of course, happens not so often, or it is more difficult. And this was investigated in the past, and it is very well known for all who know about evolutionary models that that there are differences in these possible exchanging of nucleotides. So when what I just said, this A to G, so guanine to adenine, is happening more easily, for example, than A to C or A to T. So A to G happens more easily than A to T or A to C. And this kind of mutations from A to G, for example, here within these pyrimidine or purine families, this is called in transitions, happening more easily, whereas an exchange for purines and pyrimidines, an exchange between these 2 classes does not happen so easily. And that's then called transversions. So you have here the red possibilities are the transversions, what can transform into what. Transversions are more difficult. Transitions, that's these blue ones here within purines, within pyrimidines, this is happening more easily. And therefore, when you have a mutation like here from A to G, we have this here, this situation, A to G, this is a transition, can happen more easily. We have here a T to C, can also happen more easily between these 2 specimens. That's not a problem. So again, this is also a transition and not a transversion. So we have here, let's see, A and T, A and T. And this is actually then these are all transitions. And here between this T and A, we have a transversion. So this actually would count higher. A transversion would count higher than a transition like this. So transitions are more likely than transversions. And this is now where the computer programs, the algorithms that we are using for making a tree, they consider this, whether they see in an example in a homology position like this one, whether they see a transversion or a transition. there are also further things that these algorithms can consider. So for example, this site heterogeneity. Here, I wanted to show you that there are transversions again between A and G here on this position here. This is also a transition, sorry, so that's easy, but here we have a transversion. Also on this side, we have a transversion and a transition, but usually we might have more transitions or we might have more transversions according to the age of the groups we are talking about. And this, of course, needs to be told to the algorithms. They have to consider it. Also, what we see in these alignments is that there are conservative sites, invariable sites. There's no change at all here. And when we have lots of these invariable sites, then also the algorithm have to take this into consideration. This is the number of invariable sites. Another thing is that we see, for example, when we have amino acids, let's say protein coding or ribosomal coding, the base frequencies is changing too. Sometimes we have more CG content than an AT content, and that can be really much higher. We will see this later on in an example. So all these differences in an alignment is considered and in an evolutionary model. So when your alignment here in these situations is not, is really bad because you did not care about the alignment, then you have maybe much more transversions than transitions. And then, of course, it's using a wrong model, and then the reconstruction might be wrong. So therefore, again, I want to tell you here, the alignment, check your alignments, look whether they are okay before you go in the tree reconstruction. Okay, so we need evolutionary models that differ for genes and they differ for taxa. And there are many out there that can be used. So these are only now 3 different basic models. They have abbreviations, and when you go into these different programs, including bioedit, when you do a tree reconstruction, you can do this quick and dirty in bioedit, but then you see sometimes these abbreviations, and they provide information about a certain model. So this, for example, is the Hughes-Cantor. The Hughes-Cantor is one of the eldest models, and it just counts for whether you have transitions or transversions. So that's a very easy model, actually. But then we have, for example, the Kimura 2-parameter model, and that allows for unequal transitions and transversion substitutions. So substitution means actually there's a mutation. There is one nucleotide substituted by another. That's about the substitutions. So here in this model, we can have an unequal weight of transitions and transversions. They are not the same. Here they assume in your alignment, you have exactly the same transversions and transitions. Even more complicated is when you go from here, you have now the unequal transitions and transversion solutions. And now you allow also for unequal base frequencies. So what I said about more T's and more G's, for example, in there, or C's actually, cytosine and guanine, allows for unequal base frequencies. Then you would come to this here, oh, now I forgot the name, HQY. This is also named after persons who developed this model. And now the next possibility would be allow for all 6 base substitutions to vary, and it's also reversible. So maybe you have a position where you have an A going into a T, and now here it's allowed to go back from the T to R. So this means this R, this R means actually stands for reversible, so that you also have the possibility to reverse this. The name of this one is the, oh, that's wrong, sorry, generalized time reversible model. That's actually one with a lot of assumptions, allows a lot of freedoms for the nucleotides to vary. And this is one that is taken or used a lot in these, considered a lot in the phylogenetic reconstructions. It has to be mentioned which kind of model you are using for your phylogenetic tree, which is then actually calculated. You have to mention this also in your publications. And then when you go in the publications, you will see this very often. There are other possibilities here. For example, when we start with the Hughes-Cantor model, again, it allows here for unequal base frequencies, but it does not allow for unequal transitions and transversions. Then you would end up with this Felsenstein, it's also a person, a very old one, 81. That is also still implemented, as far as I know, in some of the programs, but it's not giving enough freedom to the nucleotides to vary. Then you come along here for allow for unequal transitions and transversions. Then you come back to this model again. But in total, there are now 88 models available. If you go to, there is a book, my husband has written this book in 2005. I'm sorry, there's this line that I cannot get rid of. Okay, you see, my husband has actually written a book. It's Phylogenetic Systematics. You can go in the internet and download it. It's now for free. He paid for it so that you can download it for free. And then you will see 2 pages that explain actually these different programs. And they show exactly what they are doing. So it starts with the most primitive model with the Hughes-Cantor, and then it allows for different things, and it tells you also how it then goes on. So we start with the Hughes-Cantor, everything is actually the same. Then you have, for example, no invariable sites, and the computer programs do this. They really look, do they have invariable sites or not? If there are no invariable sites, it would end up with this situation. If it ends up with no invariable sites, then it has to go to different models. So if there is no invariable site, it would go to this. If there are invariable sites, it would still use this one, but it has to also consider all the other ones. And you see, the more you have still the basic ones like K81 or K80, this is an even elder one, or here K81. And then it looks for the other situations that the algorithms are finding, and then actually it decides which model it takes. Oh, it's frozen. Okay. So different, and now it's also now important to understand that when you are using tree reconstruction programs, they use different algorithms to calculate the best model. So there are, of course, also different programs out there, and you can do this just with the programs and find the correct, the model that fits best your alignment. There's, for example, Model Finder is a program. There's Gmodel. There is Model Test. So they are using different algorithms, and that's why they also come sometimes to different models they are applying. So again, it is also important to mention in the publication which model was then actually found. When you say the model was produced in a certain tree reconstruction program, then everybody knows which, or should know, which calculation or which algorithms were used to find the model. So you see, this is a little bit complicated. So as I said, there are programs to find the correct model that suits best for your data. And they are using different algorithms, different calculations, and these are the ones. But what is now good for us is that very often the phylogenetic programs, they have these model finding algorithms already implemented in this. So you don't have to do this separately. In former times, we had to do this separately, but now they are implemented in the programs, and they are doing this for you. Okay, so this was about finding the correct model. And now we have to talk about the tree finding algorithms. Now it's really the tree. There are different approaches, actually three different major approaches. The parsimony-based analysis is usually without evolutionary models, and it's usually only used for morphological data. When you run an analysis for molecular data and it's saying parsimony, the reviewers will reject it because parsimony is not using any evolutionary model. It will just see what is the same and what differs and will count this, and that's it. This is not correct, definitely not correct. The Bayesian analysis is a completely different situation. Here, the algorithm is actually forming all the time different, as far as I remember, because these are really complicated mathematical models. There's a lot of informatics behind that. But they're using also different kind of alignments they create by themselves, and always with a tree go back to your alignment and see how good fits the alignment, the artificial alignment on your alignment, and whether this tree might be correct. Therefore, it starts in the beginning with lots of trees that are just crap, they cannot be used, and then by doing more trees and trees and trees and trees with more analysis, it comes up to a kind of peak where they have then much better trees that better fit your alignment, and then it goes on and refines and refines. But it creates millions of trees, and in the beginning you have actually the so-called burn-in to take out. There are programs like this. Some, when you write a publication about phylogeny, there are a lot of reviewers and journals, they also still want this. But the maximum likelihood analysis, they are actually the ones that are used are the most common used ones in reconstructing phylogenies. And they just, they create trees also by millions, and they see how matches the tree on the alignment best, and then finally finds the best trees that match best with your alignment. So the program creates a tree from your sequences, and it uses then an evolutionary model that it has also calculated. So then it analyzes, that's for the maximum likelihood analysis now, then analyzes the probability or the likelihood of this tree, whether it fits your alignment. Yeah. So your alignment produces a certain tree, certain trees, usually several, and it produces a just per random a tree and looks whether it comes up with your alignment. And because it does it also millions of times, it can show which of the trees fit best to your alignment, and then can also see the incongruence between the trees that also fit your alignment. So there might be a lot of trees that fit your alignment, especially when you have not a lot of information in your alignment. So this process is done by choosing all taxa and tree combinations in different evolutionary models too. And the tree that best fit your alignment with the highest probability is chosen as final. So it's the numbers that you then see on the tree are probability values. And the conflict in the branching, the probability of the branching is shown. So the values are usually then provided. And this has nothing to do with the bootstrap. We are talking about bootstrap later on. So these maximum likelihood algorithms, not Bayes and not parsimony. The maximum likelihood algorithms are implemented in Mega and also BioEdit, but in these two programs that you know from editing the sequences now, there are only very few models are available, and you have to choose them. So they don't calculate them for you. So it might be that you choose a wrong model or not sophisticated enough. Furthermore, the algorithms are not as exhaustive. When you run a BioEdit phylogeny, you will find the tree within seconds. When you do it in a proper alignment, it might proper tree reconstruction, it might take hours. Yeah, so the algorithms are not as exhaustive, and you have only highly reduced options. So when we are using the tree reconstruction programs, IQ tree, and that's the one we will talk about later on, or WaxML, they are much better. IQ3 is the newest one. It's also working online. This one is also working online. So you just go to the server. It's in Vienna, the server. You go on that, and then you can upload your data. I will show you later on. So, and XML is working similar. So it has separate programs to reconstruct the phylogenies. It's using more sophisticated, so they are separate programs. They are using more sophisticated algorithms. They have more exhaustive model selections and search functions. And so they are more exhaustive in tree reconstruction. So when you want to publish a tree, you cannot use this. A good journal will always reject this and say, please run your tree analysis in a proper tree reconstruction program. Okay, now let's start with IQ3. As I said, you can use it online. This is actually now the online on Vienna, in Vienna, in Austria. They have this on their website, and it's easy to access it. And but you can also download the program if you have problems with the internet. Or you might, there are more sophisticated methods even in this when you download it, and therefore if you are really bioinformatician, you would probably download it. We are usually using it on the web server. So when you open it, this IQ3.org, then you see the web page, and you see also that there are many more different forms that you can do with the program. So we will use the tree reconstruction part only, but you can also use it for separately finding the correct model selection. Yeah, you can do this separately in here. Bootstrap support, it gives you also more. That's actually, we have to talk about later on again. This helps you to see whether the tree that was reconstructed in IQ3 is really very well supported, or do you have a lot of incongruence in your data? So this is a supporting program. And when you're working on genomes, for example, you can also go in IQ3 features. Okay, I said I run it on the web server, and when you open this on the web server, you will see this page. And now I will inform you about how to upload the data. What do you have to consider when you upload your data? So first of all, you have to browse in your files the alignment, the final alignment that you have worked on, and that should be really of high quality. Now you browse, you click on it, and then it will show up here the name that you have given to it. You don't have to consider all the other things actually. So you could use an example alignment just to run the program to see how it works, but that you don't have to do, of course. So you have your own alignment already here. The sequence type, it tells, is the default situations auto-detect, and they usually will find whether you have a protein, because then you have the amino acids and not the 4 nucleotides. But you can tell him also DNA if you know, well, you have the DNA. Protein is if you're working on amino acid levels, or if the codon level, it depends what you are working on. But what you probably mostly do will auto-detect, or you click on DNA. Partition file you will not include. This would be a possibility to tell the program that you have already partitions, for example, gene-wise, that you separate here already CO1 from 16S. Substitution model, you usually would say auto. Auto means that it runs it by itself. If you have done a separate model finding, used a model finding program, then you can tell this already by clicking down there. I show later on. Free weight heterogeneity, you can leave it actually, because this will be then done automatically with the program that it is chosen. Assertment bias correction, you don't have to do this. This is not really important. Branch support, we will talk later on. This you just leave as it is, because these are the best parameters that were run by a lot of simulations. Therefore, you just leave this on it. You have to include, of course, your email address, but you don't have to, because you can also leave it open, and then it will show up on your window. Okay, and that's it. Then you submit your job. So browse, and everything you can leave as it is. If you don't want to have a branch support just for a quick one, you just say none, then this will go away. And then you submit the job. For your final analysis, we need also branch support analysis, and I will talk about this later. Okay, you just, in short, again, you browse, put the file in there, you choose then DNA. Oh, I forgot to tell, if you want to upload your file, it has to be faster. It should, you have worked with faster, and that's fine. You can also use filelib. As I said in 2 weeks ago, I think there are different formats of your alignment possibilities, like the filelib or like Nexus. And that's why it's really bad working all the time with Mega. Mega is not working here. So if you have worked in Mega the last time and saved it, not as faster, but then you will see in your files that this is saved as Mega, you have to transform it first. So Mega will not work. And now the substitution model options, we just set your auto. And then when you click on it, you see I went already down, down, down. So you have many more up there. And the last one is actually the GTR. And it will then use, very often you will see GTR, the VM. That's also a very common one. And with these maximum iterations and minimum correlation, it will also find then the invariable sites and all the other stuff that needs to be included. The free weight heterogeneity, for example, it will find it. Okay, the branch support analysis we will talk later on. And now, so upload the file with the aligned sequences, include your email address, and then submit. And then later on, you will have, it will open this file, you will have a summary. And when it is still running, it will say actually running, or there will be an error maybe when you used Mega, for example, then there will be an error. You have the summary, then there's one log that tells you actually also about the models, and then you have the full results here. And the full results you can open, there's a lot of information here. Maybe to learn, you should read it, that you have an idea what is happening here. So we have here the file name. I uploaded Tambia, 3 genes. We worked on this last week. It used the model finder to find the best model and the tree reconstruction. This says something about the citation you have to use. And here you see the model finder found the best fit model according to the Bayesian informatic criterion. There are 2, but you don't, the Heike information criterion and the Bayesian information criterion, they sometimes differ, but the program will choose the one, then you don't have to do this. So in this, the Bayesian one, choose this complicated model. And when we go down, then we have here the Heike information criterion and used a different one. And then, but it used actually the Bayesian one, as I just shown to you. So it says the substitution process, model of substitution. Then it shows you the weight parameter. And you see here that the mutation weights, the substitution weights was highest towards from A to G. So actually transitions, they are, you see here the number 12. Whereas to A to C down here is only 0.13. And also the mutation rate between C and T, transition again, was highest. So most mutations, most substitutions were actually transitions. And what it also tells you, and that's sometimes of interest too, it's the state frequencies. We have here the highest rate for the T, it's 0.3. Then we have the weight matrix. Actually, it tells you here about model of weight heterogeneity, actually about the amount of no variation at all. And then we see we have more than 60% of the alignment shows no variation at all. So I am not so firm in all these data because I'm also only a user, but if you are a bioinformatician, it's really worth going in this into more detail to understand what is also sometimes happening in your tree, especially when you have a bias here towards a much higher weight of here, first of all, if you have a much higher weight of CTs, this might be no, this is not a problem, but if you have different amount of C's and T's, when you have, for example, mainly CTs in your alignment, then there might be a problem also in the tree reconstruction. And then when you go down, then you can see actually the tree. It does not look so nice. And here it's important to see a little bit no information in the alignment with regard to these 3 nodes. So 8 near 0 internal branches should be treated with caution. So it could not find a solution there. So although the tree will always be a resolved tree, there will be actually no resolution there, and therefore you have a 0 like here. 100 is very good, and 73 is already, yeah, the probability is already rather low. And what is also very important, because a lot of people do not understand this, the tree is actually unrooted. Although outgroup taxon D, that's the accession number actually. So this is this taxon actually up here is then chosen as a outgroup. It's drawn as an outgroup. It's drawn at root, and that means that you really have to be careful, because this is not your root. It was artificially done, but the tree reconstruction here is actually not a tree. It is just an unrooted relationship. And we have to talk now, actually, we have to talk later on that without an outgroup, groups that are closely related to the ones that you are interested in, that's what we call an in-group, and choosing an outgroup is very important. Yeah, let's talk about the rooting. What is a root and why is it important? Just to remember that we try to find in the tree reconstructions the monophyla that can be defined, like here A and B is a monophyleum, A, B and C is a monophyleum, D and E is a monophyleum. Remember that C and D would not be a monophyleum. So the monophyleum includes the common ancestor, the stem line, and all descendants. This is very important. And then characters that have developed during the evolution provide evidences for the presence of monophyla. These can be genetic data. It can also be morphological data. So we should have here something that defines actually this overall monophyleum. There should be a new character. And now I have put here the stem lines or ancestors, the stem lines in different colors. Here we have here blue, and this is the stem line. The red is the stem line for A, B, and C. The green is only the stem line for A and B. And what we actually reconstruct in this IQ tree is a situation like this. We do not have really a root. This cannot be reconstructed because we do not have data from this. It can only be assumed what it is. Whereas these stem lines can be reconstructed because we have all these terminal taxa here. So what the program is doing, it's reconstructing it like this. And that means we have D and E are separated by this long branch from the other 3 here, from this one here, these 3. So this line actually is a combination of this line to this separation node and this one. Yeah, it does not know where this separation actually occurs. And now the routing system. The root is now set artificially by the program. So actually we want to have it maybe here because we know from morphological character that it is here. Yeah, we have done this we know from morphological characters, yeah, that it has to be like that. But the tree reconstructing program might set it actually here. That means when you now transform this into a tree reconstruction, then you have here the stem line here along E. Then you have this situation here going to D. Then you have this and this line going then to this monophyleum. And then here you have the green line going to A and B. And you can see that this tree looks completely different from that one. So artificially setting a root changes your tree reconstruction and information about it a lot. And to avoid this situation that the program actually decides what is the root, because you want to have a tree like this. To avoid a situation like this, you use groups that are outside of your so-called in-group, but they should be closely related to this. So the arbitrary routing can lead to wrong trees and false hypotheses. So you have to avoid this. And just showing this in this situation again, and you will see this in your tree reconstruction too, because it will show up. So the tree is unrooted, also outgroup taxon is drawn at root, and that's actually this one up here. So this is the artificial root. And that makes, for example, because you might remember we used Polycera specimens and we used Tambia specimens. Because of this wrong routing, so-called wrong routing, now this Polycera is not monophyletic anymore. Yeah, we have here a Tambia in between here. So we have to choose an outgroup, and this outgroup should be closely related to your in-group. It should comprise several taxa, and it should comprise the same gene sets as the in-group. Sorry for the mistake here. It's only one R. So should comprise same gene sets. When you have worked here only with CO1, of course, you need in the outgroup also only CO1. When you have worked with CO1, 16S, and H3, you should also include in your outgroup H3. Of course, since you choose deliberately this then outgroup, it is not that big problem if you have not the same gene data set. But the thing is, what I've experienced now, it could also be that the animals you want to show where they belong to do not belong to your in-group, but actually belong to your outgroup. So the less you know about the groups here, the bigger the outgroup should actually be, so that you realize that what you think is your in-group is really your in-group and not belongs partly to the outgroup. So it's always good to have more outgroups, not really closely related. The outgroup should be closely related to your in-group, but can comprise also different groups that are closely related to the in-group. So in this case, we used Polycera specimens where we had Ferensis, we had the Hedgepetty, we had Atra and Alave. So I did not use only one species, I used several species. We could have also included other closely related polycerids, for example, Nembrota, then we would be even more on the more safe side. So we do have here already non-related species that are not related to Tambia, and we can use Polycera as our outgroup. Okay, so again, we did not specify a root, therefore a root is depicted arbitrarily. We addressed it already. So the next step is now that you have all the data, you can control this, but you can also download them right away. And then you have the download selected. It's up down here. You download it, click on it, and then you have to save them on a certain place. I called it in this case Tambia project Tambia IQ3 results. And I also call them 3 genes because these were with the H3, 16S, and CO1 that you have then the information about it, what you actually have done. So that's the files that you download. And you see here already certain icons. This is according to what kind of program you are using to show the trees. Let's see. And then we have to, next step is actually now how to show the tree, what programs you can use. And I think it is 9 o'clock. I think we just make a pause before we go now to the programs, how to show the trees. Okay. I would then stop the recording. And just see.
We will have a break for 5 or 10 minutes.
Yeah, 10 minutes. Yeah.
Okay.
And then before we go on. But is there an immediate question, of course?
Do not hesitate to asking the question. Okay,
Yeah.
folks, we have a break for 10 minutes.
10 minutes. Okay, see you then. I have to. Oh no, that's. Where was it? Yeah. Okay, so if you want to record it now, can you see it?
Okay, already live on YouTube.
Okay, so it's now at the moment, it's not recorded at all. Do you Freigabe unterbrechen? No, that's not it. Do you see still the screen?
Yes, we can see the screen.
Okay.
Please continue.
Now I record it again, but it's not in YouTube, I think. Okay. Yeah, so now the question is, how can you show your tree in a little bit nicer way so that you can start to analyze your results? And for this, there are several tree programs that to show a tree, and I will explain here now Dendroscope. Dendroscope. Another one is also Fig Tree. Fig Tree has a little bit more features, but when you have learned how to use Dendroscope, then you will also learn about Fig Tree. So what you do is this program has to be downloaded. Yeah, it's created by Daniel Hewson. So I have here rather latest version. It asks you to update it on a regular basis. And then when you have uploaded it or downloaded it to your computer, you can use it. So this is then how it looks like. You also have to register, by the way, but there's no, it's freeware. You can just register and that's it. So you go here as usual to file, and then you have to look for the file where you have your IQ tree results. So this is now our Tambia project here, IQ tree. I saved it as this file from you remember these 4 files from IQ tree. And when you want to open now, the problem is that Dendroscope does not recognize the files that you got from IQ tree. So that means when you have, this is now the one that you want to open, you see here that the files of the types that it usually sees is a special file from Dendroscope, Nexus, Newick, or NexML, or text file, but you still can't see it. So you have to click on all files. So if you can't see anything in your file where there should be these in your main file where there should be actually 4 items and there's nothing in there, then click on all files and then it will become visible. So I clicked on that, all files, and now I see these 3, sorry, not 4, 3 files. There's this IQ tree. There's also the log file that we did not look at in the original version. And here's the tree file. And this tree file you have to click on. We will open now. Then it will ask you about the internal nodes because you have these different kind of information like these support values or the information about the probabilities. And then you can say interpret as node labels, but you can also interpret as edge labels such as bootstrap values. We'll talk about this later on. The edge labels is, yeah, it does not make any difference. So you choose one. And then you see here the problem that we had because we did not use an outgroup, a real outgroup. It will show you this one again as our outgroup. And so it's selected, the selected outgroup according to the first sequence in your alignment. And this is now the tree. the maximum likelihood tree based on 3 concatenated partial genes, CO1, 16S, and H3. But this is not the final version. You see that they are all at the same end, and you see here the branches are in quite similar length. But there are several options that we also have to change here. But first of all is, of course, we want to root it according to our outgroup. So you remember that I said we had Polycera, there's 4 species as outgroup. We have chosen this as outgroup. Polycera is not Thambia. By the way, someone has his microphone. Please please shut down your microphone. Okay, how do you do this? So adjustments to be made in the tree, showing the genetic distance we have to talk about, rooting the tree with the proper outgroup, and bringing the outgroup to the bottom so that you really can finally interpret the tree. So we do first showing the genetic distance. This is actually this button we have to click on. The next thing is with the rooting, I show you here. And the third is bringing the outgroup to the bottom. That's actually here, what we call ladderized left. Just to explain a little bit more here, you can try these features. This is most important is actually to bring your alignment, make it a little bit larger or a little bit smaller here. These are things that are of interest, but most important things are actually here. Okay, so I clicked now on this button that shows you the genetic distance, actually a little bit the substitution or mutations between the sequences. And suddenly you see that your outgroup, what was meant to be an outgroup, is suddenly up down here. So you have completely different lengths of the branches. And then what you also see that Polycera is so long, so far away here. So there are a lot of substitutions that differs this from all the other Thambias. But you also see here already, there's actually no distance between all these Brasilienses here. Yeah, they are actually quite the same. So with clicking on the genetic distance, you have already an information about, how much information was in your alignment to form this monophyleum, because this would be a monophyleum. But we have not rooted yet. So before you start to interpret, next step would be to root it. I just forgot to tell you here, this is also of importance because it tells you the substitution weight that is actually calculated by the substitution models that you used. And you see that this tiny line actually means 1% of difference in the overall length of your alignment. That means when you have 1,500 base pairs, the length of your alignment, then 1% is 15 mutations. So around 15 mutations. So this length would mean 15 mutations. So you would have 15, 30, 45 mutations that differs this clade at the moment from this clade. Okay, but now we really want to root. We want to root here at the base because we have here Polycera and here is this Polycera in there, and Thambia, we will see where it comes out. There might be a problem here, maybe a misidentification. So we will root here. That means we click on this line. We click on this line here. So we click on that. And you see now it's highlighted, and also the background will go green. I don't know why they did it like this, but now it's light green. So you clicked on that. And the next step is you click on this, or you select. You can also select root. So when you click on this, it will then put this actually as an outgroup. So when the branch that you click on unites then the overall outgroup. So we did this, and then you see it rearranges, it brings your outgroup down there. So we have the Polycera, these 2 species, and we have these 2 species, and we see that this Thambia SP comes in with a very short branch. So we said that this is an outgroup. If how to say that this is not the outgroup is a little bit difficult. I think this is a misidentification. Now to see this a little bit better, what is more related, I prefer to ladderwise left. That means we are clicking now on this button. Done. So ladderwise left, when we click on this, and now have a look, we have here the sister group to this one is of course the whole part here, but then we have the sister group of this one is here, sister group then of this one is down here. So it goes in this direction, and very often in the literature you will see it like this. And to my convenience, also I think it is more convenient to put it in this way. So we have here the outgroup, and now we have the phantasmalis down there, and then we have the Tambia Eliora down here, and so on. And now the tree is actually ready for interpretation. And now the see, we have all the Brasiliensis, they are really going together. So we had only one, only the cytochrome oxidase as information for this one here, and the same, only H3, because of the overlapping in the alignment, the overlapping of genes, it still brings these Brasiliensis all together. And that's then quite nice. That's what you want to know. What do we have here? We have here Abdere, Fusca, a new species, and Abdere. Yeah, that's now interpretation. Is this a problem in the alignment, or is this maybe a misidentification? Okay, now yeah, at least you can start to interpret the tree. And again, what I wanted to say is this line now is 1% difference of the overall length of the alignment. So it's not 1,500, it's 1,445. So this is actually 14 mutations. The length of this is 14 mutations, and it helps you to see what is really very well supported by many differences and what is less supported. So in this case, for example, there's nearly no support for uniting these ones. This is extremely short. And just to tell you here, these are the probabilities here. They are, oh no, actually this is a certain supporting evidences with a certain supporting, how to say, information that was one additionally in the IQ3 program. And actually this is something we have to talk about now, because you see that these numbers differ a lot. So whenever you have low numbers like this one here, then there are many trees. Well, this is just a kind of consensus, yeah. And there are many trees that have actually not this kind of node, where this part is sister group to this one here. But whenever you have 90, something above 90, 95 above this, this is in a very well support that, for example, in this case, this monophyleum is sister to this monophyleum. Where else do we have something like this? Let's say here that's Tambia, and with this Polycera, you see it has only 88% congruence. That means this is also not very high. Whereas this 96.4, Polycera atra is sister to Polycera alaba. That's quite for sure. The evidence, let's say, to put it in this way, the evidence is very high. Yeah, okay, let's talk about support values. And here is important, the so-called bootstrap support values, because they are used a lot in these tree reconstructions. And why do you need them actually? These values, they provide evidences that the nodes that are reconstructed by the maximum likelihood analysis or by the Bayesian analysis, that these nodes are might be correctly calculated. Of course, again, this is only evidence. This is not, it cannot be the truth. Yeah, this is just the likelihood, or how good the support is in percentage. That's why I wrote it might be correctly calculated. So what we have to consider here, the trees are, again, hypotheses based on many assumptions. So all the evolutionary models that were used are assumptions, and we do not know the truth. Again, so always the tree is an assumption, it's an hypothesis. And if we want to have further support for the nodes that we see here, we can rely on supporting evidences, but not full. Yeah. And these evidences can also be morphological characters. So you can go to your tree and see, are there morphological characters that support the nodes? But we have then the possibility to do this on a molecular base, and these can be support values based on, we have these evidences based on different approaches, including molecular data. So what can we do? Coming back to this page of the IQ tree on the web server, remember that we you put your file in here, and then we have this so-called branch support analysis. And there are 3 different possibilities in IQ tree. Major possibilities. First of all, it's the bootstrap analysis. This is the most common one that is known. Then we have the single branch test. I have to explain that, or I have to say that I am not familiar with this. This is a rather new situation here. And we have the approximate base test. You can also use this one. And I will explain a little bit more in detail the bootstrap, because this is the most common one. But when you click, and that's actually by default, you don't have to click on it. This is automatically done. This single branch test. It's called approximate likelihood ratio test, and it's Shimodera Hasegawa. That's this special approximate likelihood ratio test. So that's actually the abbreviation here. And if you want to know more about it, you really have to go into the original literature. And the approximate taste test is a very slow one, and but it's probably also a very accurate one. But then you have to wait maybe many hours or even days. Okay, but I want to explain you the bootstrap, because this is more common and more known. What is a bootstrap actually doing? We have to go back to our original alignment. And what is the program now doing? It is using this complete alignment. We have here 1,445 base pairs. We have 49 total sequences here. And what is the program now doing? It chooses randomly about 70%. I am not sure whether this is always in all programs 70%. It could also be less or a little bit more. And sometimes you can even choose the amount, but it chooses randomly, let's say 70% of all available positions. We have 1,445 positions that can be chosen. So let's say it chooses maybe these first 7 positions, then it does not choose the next ones, then it chooses 4 here, it does not choose these ones, then it chooses a little bit more like here. So this is the 70%, but it still needs to a complete alignment of 1,445 base pairs. So it completes the data set by duplicating or tripling per random some of these chosen ones. Yeah, just by random, it chooses some of them and duplicates and triplicates until it comes back to a complete alignment of 1,445. Why is this now of relevance? When you have, perchance, why it's like this, an alignment that is for example, very conservative, and it chooses per random just the conservative ones, where there's no information how the animals are more closely and less closely related, then of course the program, and then it duplicates even this part, then the program runs in the problem how to know or how to calculate now the closer relationship and where it's not closer related. So this might be a problem when you have an alignment where you do not have a lot of what we say information or phylogenetic signal in there. So in this case, it's probably not the problem. So it has done this just once. Yeah, it has created a new alignment based on your original alignment. And then it is reconstructing the tree, and then it's saving the tree. And this is tree number 1. Now it does it a second time. It's what's called a second replicate. Now it's choosing completely different per random positions. So in this case, I've just chosen these ones here, left out these ones. They are conservative anyway. taken these ones now, left out all of these ones, and so on again. So in this case, 70% would mean it has chosen 1012 available positions. And then it completes the data set again by duplicating or tripling per random some of the chosen positions. And then a second tree is reconstructed and saved. And this does it according to what you say. So per the times, how often it does this process. And usually the default situation now is 1000 replicates. When you have a data set that has not a lot of information, not a lot of phylogenetic signal, it may run then for a long time with these 1000 replicates. So might be better than only choose 100, but that's standard now, 1000 replicates. So then finally, so it does it 1000 times, then you have 1000 trees reconstructed and saved. And then it produces a consensus tree. So it looks in these 1000 trees, which nodes occur how often. And then it puts this number on it and says, okay, in 96% of these 1000 trees, it found this node. But then maybe you have a number like 50, and this 50 says that in these 1000 trees, only in 500, it found this node. In the other 500, it found something else, maybe something like this. So it tells you the tree, the consensus tree that it finally shows you. With these numbers, it shows you how reliable or how well supported the node actually is. Yeah, so these are assumptions again, of course, but in general, we say that 100% like here, let's say this node and this node were in all these 1000 trees reconstructed and found again. Yeah, so that's very well supported. And then, of course, we say, yeah, it seems that A and B is really a monophyleton. It was found in the maximum likelihood analysis, and it is supported by 100% in the bootstrap analysis, and the same here. This is now a little bit difficult. It depends on the researcher. It depends on the groups. So for example, I would say 96 is still okay. So 96% of the trees show this node. But when we have only a 50, then we can say this node is not really reliable at all. It's we have to reconsider this when we are discussing the tree. So here are bootstrap values now. Shown again in this tree that I showed you before. And then you see this 100%. This is very likely, I would say 100% supported that this Brasiliensis now is sister group to this Tambia CF tenui lineata. But of course, we see substitution weights. There are differences, of course. We have already a lower value here that Tambia cava is sister group to this monophyleton. But here the 7.5 is nothing. Yeah. So you cannot say actually that this monophyleton is sister to this one here. You cannot say that at all. Actually, you should collapse that. You should collapse that, and here you should collapse it here. Oops. And collapse it actually here, and without any information where this comes out. So 69 is also not good, and 6.7 you can actually forget. So in some situations, you can also collapse this by the programs that they actually show not this overall tree, but only the one that has at least 50% evidence of support, 50% support. Okay, how to get these data? In the IQ tree server, you have then 4 files. You have to separately download them. When you push the download button, of course, you get them all. So you get these 4 files. It's this result of the bootstrap analysis. And this is the con tree. This is the consensus tree of these 1000 ones. And these are the other 3 files that you get anyway, even if you don't run this supporting analysis. This, by the way, is the fast. Second is the faster file. It's the original faster file you use for the analysis. So if you don't find your original one where you got the tree from, it's in this file too. You have a text document with all the information about finding the best model. That's here in the text document. And this is then the tree file that we actually looked at already in the beginning, that we have worked on. And it starts with the ratio values with these approximate likelihood ratio tests values on top of the tree. Yeah, so this was the support values. And the last thing that I want to show you is that is of importance for you if you just want to know about your species, about the, how to say, whether you might have a new species or where it might belong, whether this is actually a separate species or this is just a large species with lots of intraspecific variability. And for this, you can make a kind of table where you compare the intragenetic speciation rate and the interspecific speciation rate, or distance rate. If I go back just to show this here again, you can see that here the genetic variability in Brasiliensis within this is extremely low. Yeah, when we consider that this is 1% difference, 14 mutations. So this may be only one mutation, and here we have no mutation at all. So the intraspecific variability, the genetic distance between these different Brasiliensis species is extremely low. However, when we compare this one already with the next one, with Tambia tenuilata, we see that these are about 30 substitutions and another 30 around here. So we have already a higher genetic distance between this one and that one. And this you can show then in a matrix like this. It's produced in Mega. This is already made nicely for a publication on a group of the genus Phyllidiopsis with CO1. You usually do this with the CO1 gene. You can do it also with 16S, but you should do it at least separately for the genes, not the concatenated one. And since CO1 is the barcode gene used in many ways, I would do it mainly for the CO1. And you see here, these are the different species I was looking at, and you have the same species up here again. And we had now within, for example, the Phyllidiopsis krempfi, we had about 4 or 5 specimens, and the intra-specific variability, the genetic distance, goes between 0, because some were just identical, and 2%. This is actually the percentage. It's already calculated in percentage. So we have here 2% up to less than 3% intra-specific variability in Phyllidiopsis krempfi. When we compare now Phyllidiopsis krempfi with Phyllidiopsis burni, that's another species, then we see that the minimum value between Phyllidiopsis krempfi specimens, yeah, we have here about 6 different specimens. And so we have to calculate from each specimen here to each specimen of burni. And then we see that the lowest difference between specimens of here to specimens of there was 8.65%. And the highest distance was more than 10%. Whereas when we compare the different specimens within burni, we have such a low distance with only 1.3%. So intra-specific, we have here less than 3, less than 2. When we look here in the Shirine, this is a little bit more. It's like nearly 4%. Here we have in Sphinx, we have nothing, no difference at all. And that's also less than 2 and no differences at all in Cardinalis. Well, this was only one specimen. But we see also that the highest distance between the species was here between Phyllidiopsis anne to Phyllidiopsis burni with 20%, more than 20% difference. This table helps you to see what is the general intra-specific variability and the inter-specific variability. And then it helps you when you have, for example, one specimen to see, is this really a different one, or does it come up inside? Yeah, so might be you have a misidentification and the genetic distance of this misidentified specimen lies inside, comes up in the monophylum of Krempfi, and then you can look specifically on the intraspecific variability of this, and then you see, oh yeah, it must be definitely a Krempfi. It cannot be something different, a different species, for example. Yeah, so it gives you a sense for what is the interspecific variability, and the lowest distance is here 8.65. So it's about 9%. Between these different species, there's at least 9% differences. And that's how to interpret this intra and interspecific genetic variability. And you can run this in MEGA, this distance analysis. That's why MEGA is also helpful. It has many, many features. How do you do this? You open the FASTA file, or you open this actually with the data. So file here, open it. Then you click on align, because there comes then this special window asking align or analyze. You click on align. Then the alignment opens. Yeah, when you want the alignment open, you have to click on align. Then you click on the button distance. I show this no more. You click here on the button distance. Then you provide the adequate settings. You run the distance matrix, and then you export in Excel for further working. So just in detail now. Okay, you have opened your alignment. You click on the button distance. When you click on the button distance, it tells you compute pairwise distances, compute overall mean distances, compute within group mean distance, compute between groups. So you have already a lot of possibilities. But you click on this compute pairwise distances, then you see that you have possibilities now. For this analysis, this actually opens, and now you have to tell him what kind of distance. And now hopefully you remember that these genetic distances, we used substitution models. Yeah, we actually run different kind of models to say what is more likely. And in this case, we want to have the real distance, what we call the patristic, the visible distances. So when you click on this model method, you say P distance. And this then actually counts the real the visible mutations between the two sequences and brings it in relation to the length of the overall alignment. So you click on P distances, you say OK, and then it opens this file. And this is now not really very nice, because you have lots of numbers here. And you also see that this is not in percentage yet, but now you can export it here in Excel. And then you have several functions where you can say this in percentage, or this is still not in percentage. These are then things you can run in Excel, how you want to present this in your final analysis in your publication. So I would usually transform this in percentage. So here then would be 14%. This is only 1% here. And you see when we have here the Brasiliensis again, we have here the Brasiliensis on top of this. And then you see this is the intraspecific variability between all, you see here all the Brasiliensis. So on specimen, from specimen to specimen, you see, and there are no differences at all, as I said already. And here is one difference between this, the Brasiliensis, and this Brasiliensis. It's really low. And now for the interspecific variability, again, we take Brasiliensis and now use these different other species. We have Tambia affinis, Tambia amacusani, and you see here it's 18%, 17%, 17%. This is really high again. But to make it nice that you really have a table for publication, it needs some work. You cannot publish like this. Maybe you even transfer it and do it like maximum and minimum values, and then bring these data, maximum and minimum inter and intra-specific variability. Okay, so that's it for today. And actually, I am also at the end of my lectures after 4 lecturing. I think you have the basics in really to work with this, but I also know that I might not have presented everything absolutely correctly. I have to say again, I am not a bioinformatician. I'm not an informatician. I am not familiar with the algorithms. I am a user. And so please excuse me if I have not really explained everything absolutely correctly. But using it now and interpreting the results, I hope that this is then good enough. Yeah, thank you.
Well, thank you so much, Professor Heike. We have, well, the audience is increasing somehow. So, friends and participants, maybe you have any questions, please, or suggestion. Maybe James.
Okay, Prof. Heike, to take a look about the mutation, so we need more than 1 genes to analyze, right?
Well, it always depends on the question you are asking. As I said, if you want to identify specimens, CO1 is still okay. But if you want to know more about relationships, as I said, you need more than only these mitochondrial genes. Even CO1 and 16S is not really very good to see the relationship between the species. So for the barcoding, you still need a tree, yeah. But and the distances between what I've shown last, the distances, of course, they are there. So, but it does not tell you about really the relationship, which species is more closely related to what other species. And therefore, you have to be a little bit careful with this. Therefore, you need other genes too, especially nuclear genes.
Okay, the next, the reality we always use based on the NCBI data.
Mm-hmm.
That's not good, I think, but now we have a lot of tools to decide it and to interpret.
Interpret.
Yeah.
Well, I would not say that it is not good using the NCBI data. These gene repository databases, they provide you with all information that is already available, where people have already published on. And this information, since we do not invent the wheel again and again, we use already things that are available for us as information. So to build a car is only possible because we had lots of people who have worked on these issues, how to build a car. So even if how to build a tree and how to know where your species belongs to or your specimen belongs to, you don't have to invent everything by yourself again. So that's why you have to use the repositories. That's why you have to go in there. You have to download this information because this is published work. And also this is international. You have to recognize what is already published, what kind of information are available. If you don't do this is not an international standard. Yeah.
All right.
So working on a certain group means you have to get all the literature about this group. And so what can you say about the group? Yeah. So when you find something in the reefs, when you find something in the rainforest, publishing the sequence is nice for others, they may use it, but if you really want to work on them, you have to also work on everything that is already published on this special group.
Oh, yes. Danke.
Ja, bitte.
Danke. Okay, maybe another question from the audience. Citra Simarmata, do you have any question?
I think this is.
I have something to. So, in order to choose the outgroup, so like we have to look here like for heterogeneity on we use the Johnsons or Gosliner, eh? But then if somebody of us working with the fish, so that's mean they have to look for another what is called kilogram, huh?
Yeah, of course.
Yeah.
When you work on fish, you cannot look in the literature about sea slugs. You have, of course, then to go. You have. Well, if you work on a group, you have to know already a little bit about what you are working on. Yeah, if you, for example, you have a fish, and then you download, because they look similar, all the shark stuff. Yeah, a shark is not a fish. Then you will never find out where it belongs to, the fish. So you have to know already the taxon you're working on. That means it's a fish. Now, when you look at the databases, there are thousands of data in the repositories about fish. So when you narrow down already by the morphological characters or asking an expert, then you know, of course, already, okay, this is maybe a gobiide. Yeah. And then when you have this correct, you checked it again, is this really a gobiide? Then you go into the repositories and download maybe everything what is there on gobiide. And then you can run the analysis, and then you get an idea where it might belong to. Yeah, this is still not, this is a quick analysis so that you get a better idea about what do you have in the reefs or what do you have in the rainforest. But then actually, when you know what it is, your work should start. You start, and then you see what are morphological characters. Is this really a published species already, or is this a new species? Meaning you have then to go in the morphological data too, and what is published on that group. So what I've shown to you now is just to get an idea where your animal might belong to, and for this species identification. So for a taxonomist, for an expert, this is the start of the whole thing. And if you are not an expert on the group, I would always now recommend really asking experts to participate in your projects, because then you are on the safe side.
As well as me.
Okay, danke.
You're welcome. So if there is no other question, maybe we have to close this webinar. And deep in our heart, we would like to say thank you so much, Professor Heike. It's really, really very helpful. Information very helpful, very useful. We already record the conversation or this webinar. If somebody would like to retrieve again, then we can give the link of the YouTube, the address of the YouTube. Before we close, Dr. Mehdi, we have a last word or closing remarks.
Oh, probably James will do that.
Okay, James, please.
Okay, I want to thanks to Professor Heike, where Mehdi and me attended at a museum and making some work, a lot of work, I think. And as well as you gave for us to improving my competency in marine science or in my faculty. So we hopefully should be continue, because
This collaboration.
because we need the the expert.
Yeah.
We need the expert
And
improving our competency. So we
our
thanks
competence.
for this collaboration, and hopefully we should continue
Well,
for the next.
in the
Yeah, thank
future.
Thank you.
Yeah.
Thank you for the
So,
nice words. Yes.
Yeah. Well, he will be better later on though. And Professor Heike is the only one during our time in university that always, time by time, year by year, always be with us. It's very useful. We have to thank God that he already dedicate time and knowledge for our faculty, especially study program, marine science, leading by candidate professor Medi Ompi. This is very, this is the first time that we feel and then we face that Professor Heike always be with us, improving the knowledge, also information that I have lots of collaboration with other countries, but only Professor Heike gradually year by year be with us. Thank you so much, Professor Heike. Hopefully you understand what I mean.
Yeah,
Is
I
there any
understand.
other words from Pak Franz here? Pak Franz Rondonu.
Yes, from Faculty of Agriculture. So especially my department is plant diseases, plant pests and diseases. But very interesting because virus consists of RNAs, especially satellite virus to control main virus. I think maybe Prof not about it, but I think DNA or DNA analysis is very interesting for me.
Good.
To
Thank
relate it with virus as cause plant diseases, plant disease. Just that, Professor Heike, about virus.
Thank you,
Yeah.
Franz. I didn't know that
Bill.
we have another member that from other faculty.
Yes. I invite.
Oh yes.
Oh, you invite
James.
Franz.
Yeah. Sorry. Thank you, James.
He's my best friend.
Yes.
Dr. Franz, you can develop for the collaboration with Professor Heike.
Yeah, I think very good
in the
idea.
future.
Yes.
If you would like to.
Yeah. Well, as I said again, I am not an expert in viruses of plants, and we have really to be careful with this. I can provide you a little bit of methodologies, but working with RNA is a little bit different as DNA, and I have not worked so much with RNA, so I would, you have different programs here, you have different databases here. I would be then very careful. Yeah. Of course, we can collaborate maybe on a base where I have expertise, but when it comes to final analysis, I can always recommend for all of you, you need the experts on the special field. Yeah, as I'm not an expert on genomic data, I would never collaborate with you. This is impossible. Yeah. And same, I'm not an expert in insects. I'm not an expert in insects, not an expert in plants. I would not even like to work on corals, although I'm a marine biologist. Yeah, this is, we need the taxonomist, the person who knows about the groups when you really want to know more about it and go in deeper in the field.
Yeah, correct.
Yeah,
that's different.
Yes.
But the
Thank you, Prof.
information
Okay.
you're welcome.
The information that you already given to us is really a basis for another step project in the future. And we really appreciate that, Professor Heike. Thank you, Dr. Franz.
Oke, sama-sama. Satu jemaat jadi.
Oh,
gets money ya, orang kan sama-sama dulu.
Okay,
Yeah,
thank
okay.
you,
Yeah.
Merfat.
Okay,
Dr.
then.
Billy, there will
Well-
be a chance to bring Professor Heike for next year to Manado.
Yeah,
Yeah.
maybe when she will be here, then everybody or who are interested in this field, then you can ask question. Because here we are limited of time.
Okay.
Yeah, as hopefully with Corona it is possible, and I would like to come then at the end of next year, that from October maybe on. That would be
Hopefully.
nice. Yeah,
Hopefully,
already many
Professor Heike.
obligations before that.
Oh,
Yeah,
But
very glad to
October,
hear that.
October to December, it's still free for Indonesia.
Indonesia, wow.
Yes, for
Thank you so
Means
much.
what? You're welcome.
Don't forget
Okay.
my black chocolate.
Okay, then. Thank you very much
Okay.
again,
Okay.
and
Thank you.
giving the chance, and see you hopefully next year. Not only virtual. Goodbye.
Bye,
Bye.
Professor
Bye.
Heike. Bye, Mehdi and
Bye-bye.
Dr. James Paulus.
Yeah.